Let's jump right into this explainer. Today, we're demystifying psilocybe mushrooms, and I promise you, we are going way beyond the famous psychedelic stuff. We're going to look at the structural chemistry, some really weird storage quirks, and a whole world of untapped medical potential that's quite literally hiding in plain sight.
You know how we constantly hear about their potential to treat depression and anxiety? I mean, that absolutely dominates the headlines right now. But what else is actually hiding inside these fascinating fungi? If we look strictly at the scientific data from the past 67 years, we find a story of hyper-focus on just a couple of molecules, leaving a massive, highly complex biological resource virtually unexplored.
Let's just start with the sheer scale of this. As the second largest genus in the Hymenopteraceae family, there are exactly 311 cataloged species of this mushroom globally. Think about that. 311 distinct biological factories, all pumping out their own unique, complex compounds.
And their global footprint is absolutely massive. We're not talking about some rare plant in one obscure corner of the world. They span from the warmest climates all the way to the coldest, with the highest density concentrated right in Europe and North America. This represents a globally available biological resource sitting right in our own backyards.
Which brings us to Section 1, the tip of the iceberg, the psychedelic heavyweights.
So, for over 60 years, the scientific community has been completely fixated on just the tip of this chemical iceberg. Ever since Albert Hofmann isolated them back in the 1950s, almost all the literature hyper-focuses on isolating and quantifying just two major compounds, psilocybin and its metabolite, psilocin. Because these two compounds mimic serotonin and trigger hallucinations, they naturally get all the funding and attention. But here's the crazy part. That leaves 35 other identified compounds in this genus largely ignored by science.
Now, you might be wondering why the tunnel vision. Well, a major variable driving this massive research gap is their DEA Schedule 1 classification. It's a strict regulatory status labeling them as having zero accepted medical use and a high potential for abuse. This makes widespread chemical evaluation incredibly difficult. Researchers essentially have to jump through massive legal hoops just to study the basic chemical makeup of these fungi.
Okay, let's move right into Section 2, beneath the surface, the missing metabolites.
If we manage to peel back that regulatory red tape and look past the psychedelic fame, we find a deeply complex chemical world. Beyond psilocybin and psilocin, researchers have actually found 23 other alkaloids. And some of these are truly remarkable. They act almost like molecular bodyguards. Take beta-carbolines, for example, like harmane and harmine. They actually inhibit stomach enzymes. So by blocking those specific enzymes, they essentially stop your body from destroying the other active chemicals before they can even be absorbed. It is literally a built-in chemical delivery system.
Breaking down the numbers here, in total, only 37 compounds have been formally identified across this massive genus over 67 years of study. That breaks down to 23 alkaloids, 7 terpenoids, and 7 phenolic or other compounds. Just think about that for a second. Across 311 species, we've only mapped 37 molecules. It's kind of like having a massive library of 311 books and only reading the first sentence of 37 of them. It just perfectly highlights how truly underexplored the chemical diversity of this whole genus remains.
Moving on to Section 3, myth-busting, storage, and stability. Now, the data we're looking at completely busts some massive myths about preserving these delicate compounds. I mean, you'd probably think freezing fresh mushrooms is the absolute best way to preserve them, right? Well, the data shows the exact opposite. Freezing fresh mushrooms at -20 degrees Celsius actually destroys over 80% of the psilocybin. And drying them isn't a perfect fix either. You lose over 50% of the concentration in just the first few months. Surprisingly enough, the absolute most stable method tested for retaining high concentrations for months on end is simply storing them fresh, at room temperature, around 20 degrees Celsius, in the dark. Who knew?
Let's head into Section 4, Chemistry in the Wild, the Blue Bruise.
This extreme chemical fragility we just talked about actually brings us to a really cool phenomenon known to pretty much any mushroom forager, rapid blue bruising. It happens when a psilocybe mushroom is injured, picked, or literally just bumped. The whole bruising process kicks off with a reaction called dephosphorylation. In plain English, this is a chemical process where a phosphate group is stripped away from the stable psilocybin molecule through hydrolysis. Once that phosphate is gone, boom, the molecule converts into psilocin, which is highly unstable and readily oxidizes the second it hits the air.
So here is how this chemical cascade actually unfolds in real time.
Step 1. A specific enzyme in the mushroom called phosphatase Cy goes in and aggressively removes that phosphate group. Step 2. Immediately after, another enzyme called lacase Cy steps in and oxidizes the freshly exposed psilocin. And finally, step 3. These newly oxidized, highly reactive molecules crash together, linking up to form complex, visually striking blue polymers.
And the result of that molecular crash? Dimers. These are essentially two of those oxidized psilocin units locked together at specific carbon positions. Now, you might be asking, why does the mushroom do this, especially since it's totally destructive to its own active compounds? Well, scientists hypothesize this rapid chemical explosion is actually a biological defense mechanism. It's meant to deter insect predators, like larvae or ants, that might be trying to eat the fungus. It is literal chemical warfare happening right before our eyes.
Which brings us to our final part, Section 5. The climax, untapped non-psychoactive potential. Speaking of defending against predators, this opens up what is arguably the most ignored frontier in research today. While everyone is focused squarely on mental health and serotonin receptors, tests on entire mushroom extracts have demonstrated some truly remarkable non-psychoactive abilities. We're talking about killing mosquito larvae, which, if you think about the global implications for insect-borne diseases, is huge. They also fight dangerous foodborne bacteria like E. coli and Salmonella, alongside showing significant antioxidant and anti-inflammatory potential.
But here is the absolutely crucial point. Despite knowing that the whole extract can fight bacteria and inflammation, not a single one of these physical, non-psychoactive medical benefits has been linked back to a specific, isolated compound yet. The research simply hasn't been done. This means that major pharmaceutical breakthroughs for physical illnesses could literally already be hiding right there in the metabolic data of these mushrooms.
This staggering gap is exactly why the authors of this massive review are sounding the alarm. They're practically begging the scientific community to pivot. They state we must focus efforts on isolating new compounds and evaluating other types of biological activities that can improve the knowledge of mushrooms' alternative applications. In short, we desperately need to start looking beyond the psychedelic trip.
Because as we move past this psychedelic renaissance and finally look at the vast, unexplored base of this chemical iceberg, it leaves us with one incredibly exciting question. If we have found all of this from studying just 2 molecules, what other cures are waiting to be discovered out there in the forest?